Thanks. So I've spent most of my scientific career working with neglected tropical diseases. And one of the things that's always bothered me about neglected tropical diseases is why are these diseases neglected? They affect 1 in 6 people. They affect 1 in 6 people in the world's population. They also have a very large geographical distribution. So the countries that you see highlighted in the darker blue here have at least one neglected tropical disease present. And that goes up to the countries that are in red, and they have more than 5 neglected tropical diseases present. So we would have in this room probably about 3 to 4 people infected with a neglected tropical disease if we were representative of the world's population. So if these diseases are so prevalent and have such a large geographical distribution, then why are they neglected? Well, it's because they affect the world's poorest people, and that they're also known as the bottom billion. So the bottom billion are the billion poorest people in the world. The World Health Organization is the entity that actually designates diseases as neglected or not. And here the list you see behind you is the current portfolio of neglected tropical diseases. And you have a variety of types of diseases in their portfolio. So you have some viruses, you have some guinea worms, some helminths, some single-celled eukaryotic pathogens. So you're never going to be able to do drug development on all of these at once. So our lab focuses on leishmaniasis. And you get leishmaniasis when you're bitten by an infected sandfly. That infected sandfly will deposit the parasites whenever it takes a blood meal. Those parasites will then be taken in by your cells like macrophages, and inside of those macrophages, the parasites will divide and multiply. Eventually, the macrophages will burst, releasing the parasites, and the parasites can then be taken in by another sandfly and go infect someone else. The focus of the talk today is going to be a specific type of leishmaniasis called visceral leishmaniasis or Kala-Azar. Visceral leishmaniasis is primarily prevalent in the bone marrow, spleen, and liver of patients. In fact, the spleen and liver get so infected with parasites that they swell, and you see a distended or swollen abdomen in patients with visceral leishmaniasis. Also, the mortality rate of this disease is almost 100% if you're not treated. Visceral leishmaniasis is prevalent in a diverse group of countries covering a large part of the world. The yellow dots on the map represent cases of visceral leishmaniasis, and most of the cases are actually seen in India, Bangladesh, Sudan, South Sudan, Ethiopia, and Brazil. And you can imagine that there are some treatment limitations that are present in these countries. Long hospital stays, injections, and high costs of treatment can prevent patients from actually receiving effective treatments. The current treatments on the market are pentastam, which has a long injection course, resistance, and some cardiotoxicity issues. Ambisome has a lower efficacy in some regions, is very expensive at $200 per regimen. So here, us sitting at Ohio State University in the United States, we don't think about $200 for a life-saving regimen to be cost prohibitive. But in some of the countries where leishmaniasis is prevalent, that is cost prohibitive. Ambisome is also only delivered by IV, so you can imagine, as you've seen in some of the previous pictures, that can be difficult for administration in some of these areas. Paromomycin is another current treatment. It is administered by injection for 21 days. So it's fairly affordable, but the 21-day injection cycle makes it less desirable for treatment in these areas. Miltefosine is the only orally available treatment on the market. The big problem with miltefosine is it actually causes birth defects in pregnant women. So pregnant women cannot take miltefosine at all. It has GI toxicity, and it's getting to be a little bit more on the expensive side. So what are we looking for in a treatment? We want something that's effective in India and Africa because that's where most of our cases are occurring. We want something that's active against the resistant strains. We want something that's inexpensive. And we want something that's going to be oral. And I would say that oral treatment is probably going to be one of our largest hurdles with treatments for visceral leishmaniasis, because as you could see from the previous slide, that most of the treatments are either IV or an injectable of some sort. So overcoming this oral bioavailability barrier, I think, is the greatest obstacle for meeting all of these marks. So our lab has taken a combination approach, and we have decided to use DB766, which is a compound that was identified from a larger screen. It was a good candidate, but it wasn't quite effective enough alone to be used as a single therapy. we also want to look at posaconazole. So posaconazole is an FDA-approved drug, and it's got some interesting properties in that it actually is fairly, it's not quite effective enough in the animal model alone, but it's more effective than what we would have predicted based on some previous studies. And it's got some interesting characteristics that go along with that. And so we wanted to see what happens if you take DB766 and posaconazole, mix them together, and then dose animals that have been infected with leishmaniasis. So in order to do that, so if you want to look at the slide here, we have the reduction in parasitemia here on the y-axis. And so keep in mind, this is reduction. So the larger the bar, the lower the parasitic burden in these animals. And as you decrease the dose of DB766, you decrease the efficacy of the compound. And that's called a dose response. And that's what we want to see when we give these compounds. The same thing happens with posaconazole. As you decrease the dose of posaconazole, you decrease its efficacy. Again, we're seeing a dose response. And these all are positive things that we want to see and indicative that we want to move forward. So then we also want to look at combinations of these compounds. So this entire line here, all of these groups of animals were given 75 milligrams per kilogram of DB766, which corresponds to the single dose bar here. And then this first animal was also given 30 milligrams, or this first group of animals was also given 30 milligrams per kilogram of posaconazole. The second group of animals was given 15 milligrams per kilogram of posaconazole, and the yellow bar was given 7.5 milligrams per kilogram of posaconazole. And we repeat this through 2 other dose groups. So all of these animals get 38 milligrams per kilogram of DB766 at the different and also get the different posaconazole doses. And the same thing with the last one. They get 19 milligrams per kilogram of DB766, and then each group gets its dose of posaconazole as well. And let's look at this blue bar right here in the middle. So that bar gets 38 milligrams per kilogram of DB766, which corresponds to this single dose bar here. And it gets 15 milligrams per kilogram of posaconazole, corresponding to that blue bar here. And what I want to point out to you is you see how much taller this blue bar is than either one of these monotherapies. And when you're giving combination treatments and looking at the efficacy of combination as compared to single compounds, that's what we want to see. And we saw that, as you can see, with most of our combinations. We also wanted to take a second approach. So we wanted to take, it's called a hybrid, we're calling it a hybrid approach to drug development. So we took the very interesting part of DB766, which is highlighted in blue, and the part of posaconazole that we're interested in, which is highlighted in red, and we put them together, separated by a linker, and made hybrid compounds. And we want to be able to have improved properties in the host. So some of these properties that we saw with our combination study, where when we mix the compounds together, they were better than either of them by themselves. And we also have the potential to hit multiple targets. So we still hope to hit the target for DB766 and the target for posaconazole with a single compound instead of a combination. Right now, we are in the process of testing the anti-leishmanial activity of these hybrids. We begin that by testing to see if they're effective against leishmania in our cell-based assay, and we test for toxicity against mammalian cells. And what we want is for the compounds to not be toxic to the mammalian cells, because those are cells like yours and mine, but we want them to be effective against the leishmania. So if they pass that threshold, we then test for toxicity in our mouse model and move to efficacy in our mouse model if they're not toxic. If we see a greater than 75% reduction in parasitemia, then we move on to our hamster model. So the combination slide that I just showed you, you saw a lot of those compounds had a greater than, or combinations had a greater than 75% reduction in parasitemia. So that combination has actually been selected and is moving on to our hamster model right now. As far as the hybrids go, we have currently tested 45 hybrids or produced and tested 45 hybrids. 13 of those hybrids have an IC50 that we are happy with. So an IC50 is just the concentration at which 50% of the parasites are dead in your assay. And 2 of those hybrids are currently ready for efficacy testing. I want you to keep in mind that that doesn't mean that the other 11 of the 13 aren't going to be moved forward. This is currently a moving process. I think our chemist gave me the word yesterday. I think we have like 3 more hybrids that he has ready. So we're constantly putting new compounds in the top of this funnel to get to the bottom. Some of those 13 we may go back and choose for additional testing as well. So I want to bring you back to our treatment goals. We wanted a compound that's effective in India and Africa. And all of the studies that I've shown you today, both of our cell-based assays and our animal models, use a strain of the parasites from Africa, so we're fairly confident that these parasites will be effective, at least against this African strain. We want parasites that are active against resistant strains. So if you remember back to the flow chart, our plan is to put a little sidebar by the flow chart, and after the testing in the mouse model, if they are effective in the mouse model, we can adapt our cell-based assay to resistant strains. So we have an imaging-based assay that doesn't require any type of reporter gene. And so we can take clinical isolates and run them through our cell-based assay. So then we can decide if these compounds are effective against resistant strains. We want our compounds to be inexpensive. And while we don't have a price prediction at this point, we do work, and at least our chemists work to keep the synthesis down to less than 10 steps. So you can imagine the fewer the number of steps, the less expensive the compound is going to be in the end. And like I said from the beginning, I think the largest hurdle in this target product profile here is the oral treatment. And all of the animal modeling data that I've shown you, the animals were given these treatments orally. None of these were injected or given by IV. So I just want to say thank you to all the people that actually did the work and for our funding agency. And thank you all for coming today.